Hello student, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to talk about module that is energy currency of the cell, and this has been taken from a paper Thermodynamics of Living System and Bioenergetics. So what is the objective of this module? So we will discuss about different energy currency of the cell. We will also discuss what is a utilization of ATP, GTP, UTP and any other NTPs. We will also discuss about other high energy compounds like phosphoanhydride, phosphoesters, acyl phosphate and thioester. So based on this module is divided into following section. The first one is what is the basis of energy currency, why NADP is used as a energy currency, why NTPs are used as an energy currency. Then we will discuss why NTP is giving energy, and we will also discuss why high energy compounds or high energy currency are utilized in the cell. Module is, you know that when a simple precursors like nucleotides are converted into polymers like DNA, RNA and protein, energy is required for the condensation and making a ordered sequences. So precursor for the DNA and RNA synthesis are nucleotides, triphosphates, and polymerization is result into the cleavage of phosphoanhydride linkage that is present between alpha and beta phosphate, which result into the release of pyrophosphate, that is PPI. So the molecule which transfer to the growing polymer in these reaction are adenylate such as AMP, guanylate such as GMP, cytidylate or uridylate in the case of RNA. But deoxy analogs such as TMP are utilized in the DNA synthesis. Not only that, when we have activation of amino acid for the protein synthesis, that also involves utilization of energy that is coming from ATP or GTP. In all these cases, whenever a synthesis take place, ATP or its homolog are used as a exergonic product that promotes the endergonic processes of synthesizing a polymer of a specific sequence. There are another molecules that also act as a storage of energy in the cell, which is also known as high energy compounds such as phosphoanhydride, phosphoesters, acyl phosphates and thioester. So we will also discuss the mechanism of favorable energy changes for these high energy compounds. We will also discuss why different enzyme requires different energy equivalents. In this module, we will also discuss what is the difference in the delta G and other changes in different high energy compounds. We will discuss how nucleotide triphosphates or NTPs are used as energy currency. Although we have focused on ATP as cell energy currency and donor of the phosphate group, but all other nucleoside triphosphates like GTP, UTP and CTP and all their deoxynucleotide triphosphates like DATP, DGTP, DTTP and DCTP are energetically equivalent to the ATP. But most of the enzyme uses ribose based triphosphate as an energy currency. The free energy change associated with hydrolysis of its phosphoanhydride linkage of the ATP is same for all the NTPs and DNTPs, as they have same constraints, which we will discuss in this module, because they have same constraint which is responsible for ATP production or ATP hydrolysis. During the various biological function, these other nucleotides are generated and maintained as nucleoside triphosphates by phosphate group transfer to the corresponding nucleoside diphosphates, that is known as NDCPs, and monophosphate like NMPs. For example, ATP is triphosphate, ADP is diphosphate, and AMP is monophosphate. ATP is the primary high energy phosphate compound produced during the catabolism, for example, in the glycolysis, oxidative phosphorylation, in photosynthesis, and photophosphorylation. Several enzymes carries phosphate group from ATPs and transfer to other molecules. They may take phosphate group from other NTPs. The delta G change for the hydrolysis of other NTPs are same as the ATP. Therefore, conversion of one NTP into another have delta G of 0. Therefore, they can be interconverted, or you can say, because Gibbs free energy change is 0, so they are existing in the equilibrium. Enzymes that involved in the reaction are nucleoside triphosphate adenylate kinase that convert nucleoside into the nucleotide. And you can see here is the equation shows nucleoside triphosphate when interact with the AMP, it is converted into nucleoside diphosphate and ADP. So you can say that this enzyme is involving in interconversion of different NTPs. Similarly, we have another enzyme that is nucleoside diphosphate kinases that catalyzes exchange of phosphate group between different nucleoside diphosphates. The activity of this enzyme, nucleoside diphosphate kinase, maintain an important equilibrium if you remember the TCA cycle, where GTP is produced in the TCA cycle and it is converted into the ATP with this kinase enzyme. So therefore, we have sufficient enzyme that playing role in the interconversion of NTPs. Therefore, either will produce any type of NTPs, they all are equivalent in terms of energy production, in terms of utilization, but still different enzyme require different NTPs. We will discuss it into the details in the coming section of this module. The nutrient which contain energy in low energy covalent bond that are not useful to do most of the kind of work in the cell. These low energy bonds must be translated into high energy bonds to perform a specific energetic function, and this is the what the function of NTPs or ATP is. A continuous supply of ATP is so crucial that if a protein that is used for ATP production, if it is inhibited, if it is attacked, then a person is dies within the minute. And such example, if you remember, is cyanide compound. They will inhibit cytochrome oxidase, that is complex 4, and inhibit the ATP synthesis. The cyanide inhibit by binding to the copper atom that is present in the complex 4, and this binding blocks electron transfer in mitochondria and ATP production, and therefore is going to be a lethal drug for a person who is taking cyanide. Then question come, why NTPs gives energy? So the conversion of ATP into ADP and inorganic phosphate, or in some other cases ATP into AMP and PPI, that is pyrophosphate, is very important for energy production from the ATPs. Then why this ATP produces energy? So ADP dependent reaction, ATP dependent reaction is driven by hydrolysis of ATP, and this reaction of ATP appears to be single hydrolysis reaction in which water displaces inorganic phosphate. ATP hydrolysis usually accomplishes nothing but the liberation of the heat or release of the heat, which cannot driven a chemical process in an isothermic system. It represents a two-state process in which the part of ATP molecule, that is either phosphate or pyrophosphate group or adenylate moiety or AMP, is first transferred to the substrate molecule or to a amino acid residue in the enzyme, become covalently attached to the substrate or enzyme and raising the free energy content. Then in the second step, the phosphate containing moiety transfer the first step in displace and generating the PPI and inorganic phosphate or AMP. Therefore, you can say that ATP participate covalently in the enzyme catalyzed reaction in which it contributes the energy. One such example is hexokinase, where ATP is used and its phosphate is transferred to hexokinase, and then the phosphate carried by the hexokinase is going to transfer to the glucose and glucose 6 phosphate is produced. ATP hydrolysis, as you know, is a favorable process. So, what is the reason that makes ATP hydrolysis favorable? So, the first reason, if you remember the structure of ATP, they have many negative charges, and that negative charges create electrostatic repulsion within the ATP. So, when ATP 4 minus, or simply you can say ATP, is broken into ADP and PI, then that result into reduction of electrostatic repulsion. And if there is a reduction of electrostatic repulsion, that makes charge separation. So, the hydrolysis of ATP is a favorable process. The second point is, the product of ATP hydrolysis is ADP and inorganic phosphate. So, inorganic phosphate, as it produced from ATP, get resonance stabilized, in which each of the 4 phosphate oxygen bonds have same degree of double bond character. So, once the one product, that is inorganic phosphate, is get resonance stabilized, it will enhance the stability of the product. That makes this process favorable. And the inorganic phosphate have hydrogen ion which is not permanently associated with any one of these oxygen. So, that stabilizes overall negative charge that is created during resonance of inorganic phosphate. So, the third point is, the second product which is produced by ATP hydrolysis is ADP. So, ADP 2 minus, which release because of breakdown of ATP, give its proton at physiological pH, which is pH 7, and converted into ADP 3 minus. So, according to the Leach-Italier principle, if in the reactant and product anyone is utilized in the reaction will favor toward the formation of reactant or product which is utilized in the reaction. So, according to Leach-Italier principle, when ADP 2 minus is produced, it is immediately converted into ADP 3 minus. That makes ATP hydrolysis favorable process. The fourth factor that favors ATP hydrolysis is the greater degree of solvation of the products, that is inorganic phosphate and ADP, as compared to its reactant, that is ATP, which is again a favorable process, because now your product is having more solvation as compared to its reactant, or you can say that product is more stabilized as relative to its reactant. The fifth point is, because the concentration of direct product of ATP hydrolysis like inorganic phosphate and ADP is far below the concentration at equilibrium. Therefore, the mass action favors hydrolysis of reaction in the cell. In this figure, as I have discussed, what are the different factors that is responsible for the ATP production? You can clearly visualize those factors. In this figure, you can see that there are presence of ATP4 minus with 4 negative charge. 4 negative oxygen have negative charge. So they have repulsion. So once this ATP4 minus is hydrolyzed into ADP2 minus and inorganic phosphate, you can clearly see that inorganic phosphate get resonance stabilized. At the same time, ATP4 minus when converted into ADP2 minus, this ADP2 minus in physiological condition donating its proton and become ADP3 minus. So therefore you can see that repulsion is reduced after ATP hydrolysis, inorganic phosphate get resonance stabilized and ADP2 minus is converted to ADP3 minus. At the same time, although you cannot see here, but the product is 2, so the solvation layer is more. So you can say the solvation energy also basically is more for the product. So product are more better solvated than reactant. So all these four factor makes this ATP hydrolysis. Although the hydrolysis of ATP is highly exergonic. When you are saying exergonic, that means heat is elaborated from this. Heat is released from this. With a delta G of minus 30.5 kilojoule per mole. The molecule of ATP is kinetically stable at pH 7 because the activation energy for ATP hydrolysis is relatively high. Therefore, it cannot spontaneously release its heat until we require. So, repeat cleavage of phosphoanhydride bond occur only when it is present in the active site of enzyme or it is catalyzed by the enzyme. This shows that being an exergonic reaction, still we will not have a spontaneous cleavage of this ATP. That basically helps our body to use the ATP in an effective manner. The free energy change of ATP hydrolysis is minus 30.5 kilojoule per mole under standard condition, but the actual free energy of hydrolysis in the living cell is very different because the cellular concentration of ATP, ADP and PI are not identical, or you can say they are much lower than 1 moles as present in the standard condition. Furthermore, the magnesium in the cytosol binds to ADP and ATP, and for most enzymatic reaction that involve ATP as a phosphate group donor, the true substrate is not ATP ADP alone, but it is a magnesium ATP complex, or you can say magnesium ATP 2 minus, because out of the 4 minus of ATP, 2 is nullified by 2 positive charge of magnesium. So the relevant delta G for magnesium ATP complex hydrolysis in the intact cell is denoted by another factor that is known as delta Gp or phosphorylation potential, and it has a value more negative to the delta G of ATP hydrolysis. The question come in our mind, why it is more negative as compared to ATP hydrolysis alone? The first point that is come in our mind that magnesium may stabilizes negative charge of ATP4 minus, but magnesium also stabilizes phosphate, magnesium also stabilizes ADP, and magnesium also increases the solvation layer. So out of the 4 factors, it is the first factor that get reduced in the presence of magnesium, but the remaining 3 factors are going to be enhanced in the presence of magnesium. Therefore, the presence of magnesium, the ATP hydrolysis is relatively more favorable as compared to its absence. Some process directly hydrolyzes ATP or GTP. For example, non-covalent binding of ATP or GTP followed by hydrolysis to ADP into or GTP and PI can provide energy to cycle some protein between two conformations and its mechanical movement. This occurs in muscle contraction, in the movement of enzyme along with the DNA and ribosome along with messenger RNA. The energy dependent reaction catalyzed by helicases, RAK protein and some topoisomerases also involve direct hydrolysis of phosphoanhydride bond of NTPs. The GTP binding protein that act in signaling pathway directly hydrolyzing GTP to drive the conformation changes that terminates signal triggered by hormones or other extracellular factors. The phosphate compounds found in the living organism can be divided somewhat arbitrarily into the true group based on the standard free energy of the hydrolysis. The high energy compound which is having delta G more negative than minus 25 kilojoule per mole or low energy compound which is having less delta G negative. The breaking of all chemical bonds require an input of energy. The free energy released by hydrolysis of phosphate compound does not come from a specific bond that is broken. It results from the product of the reaction having a lower free energy content than the reactant. That means you can say that your reactant having more energy and product have less energy. So what will happen with the additional energy? So that additional energy is released in the form of heat, is released in the form of delta G. So for the simplicity, we can sometimes use the term high energy phosphate compound when referring to the ATP or other phosphate compound with large negative standard free energy of hydrolysis. As in the evident from additivity of free energy changes of sequential reaction, any phosphorylated compound can be synthesized by its coupling to the breakdown of other phosphorylated compound with more negative energy of the hydrolysis. And this is a basis of a coupled reaction where a reaction which is not possible, that is endergonic, can be made possible by its coupling with an exergonic counterpart. Although all the NTPs have its delta G same, but different enzymes require different NTPs because of differences in the NTP binding pocket that is present in the enzyme. After discussion of NTPs as energy currency and why they are giving energy, let's have discuss other high energy compounds. So along with the phosphoanhydride, we also have phosphoester linkage containing compounds like phosphoenolpyruvate, acyl phosphate like 1,3-bisphosphoglycerate and thioesters like acetyl coenzyme. In all these phosphate releasing reactions like ATP, 1,3-bisphosphoglycerate, phosphoenolpyruvate and phosphocreatine, there are several resonance forms available for inorganic phosphate and its stability as relative to its reactant, which contributing to its large negative free energy chain. So in general way you can say that although in most of the cases it is a phosphate that release, but if the phosphate is not released, the product which is released is going to have more resonance stability that favors this large negative free energy chain for this high energy compound. The first high energy compounds other than the NTP is phosphocreatine. In phosphocreatine, as you can see in the structure, they have phosphate oxygen nitrogen bond that can be hydrolyzed to generate free creatine and inorganic phosphate. The release of phosphate and resonance stabilization of creatine makes the reaction favorable. Another word, you can say that the phosphocreatine produces creatine and phosphate. It is well known that phosphate undergoes resonance stabilization, but here the second product, that is creatine, is also undergoes resonance stability. So both the product are resonance stabilized. Therefore, the free energy chains for phosphocreatine hydrolysis is large and negative, and they have minus 43 kilojoule per mole. The muscle of the body function by using ATP for muscle contraction and relaxation. When a molecule of ATP is used in the contraction process, it is hydrolyzed to ADP, an inorganic phosphate. The muscle have limited supply of ATP, which is quickly used for muscle activity. So they need to generate ATP. One way to generate ATP is the use of creatine phosphate or phosphocreatine, which is a stored high energy compound in the muscles. So the phosphocreatine, how it is created in the muscles? So the phosphocreatine is formed from part of three amino acids. The first is arginine, second is glycine, and third is methionine. It can be synthesized by formation of guanoacetate from arginine and glycine in the kidney, followed by methylation by S-adenosylmethionine to creatine in the liver, and phosphorylation by creatine kinase, where ATP is required, to phosphocreatine in the muscle. And this phosphocreatine is synthesized in the liver, is transported to the muscle cell via bloodstream for its storage, and creatine phosphate shuttle facilitate transport of this high energy phosphate from mitochondria. It can anaerobically donate phosphate group to ADP to form ATP during the first 2 to 7 seconds, followed by intense muscular or neuronal effect. Therefore, you can say that phosphocreatine is important for muscle function. The excessive ATP can be used during the period of low effect to convert creatine to phosphocreatine. The reversible phosphorylation of creatine is catalyzed by enzyme creatine kinase. So the presence of creatine kinase in blood plasma is indicative of tissue damage and is used in the diagnostic of muscular dystrophy such as myocardial infarction when it will take place in the heart muscle. So you will know that creatine kinase have multiple forms. So some form is found in the brain, some found in the muscles, some in the heart. So based on the isoform, we can identify, we can diagnose, or we can use this as a clinical marker for myocardial infarction. The cell ability to generate phosphocreatine from excess ATP during the rest, as well as its use of phosphocreatine for quick generation of ATP during intensive activity, provide it as a spatial and temporal buffer of ATP concentration in the muscles. So in other words, you can say that phosphocreatine acts as a high energy resource in the couple reaction. The energy given from donating the phosphate to generate other compound, in this case ATP. And the phosphocreatine play a very important role in tissue that have high and fluctuating energy demand such as muscle and brain. So other high energy compound is phosphoenolpyruvate. So phosphoenolpyruvate contain a phosphate ester bond that undergoes hydrolysis to yield enol form of pyruvate, and directly product of this reaction is tautomerized to more stable keto form of the pyruvate. So because this reactant has only one form, that is enol, and product have two form, therefore the pyruvate undergoes resonance stability because of tautomerization. So as I told you, for other high energy compound, here phosphoenolpyruvate produces inorganic phosphate and the pyruvate both get resonance stabilized. So they will produce high energy. Not only that, phosphoenolpyruvate is going to have highest Gibbs free energy change if you compare it with the other high energy compound. This comes because of direct conversion of pyruvate into the keto form of the pyruvate, enol form of pyruvate into keto form of the pyruvate, and resonance stability of inorganic phosphate. After discussion of other high energy compound, let's discuss the high energy compound, that is 1,3-bisphosphoglycerate. So this 1,3-bisphosphoglycerate contain an anhydride bond between carboxylic group at C1 and phosphoric acid. So during the hydrolysis, this phosphate is removed or released from the carboxylic group, and this have a delta G of minus 49.3 kilojoule per mole. So what happens in this reaction when H2O is added across the anhydride bond of 1,3-bisphosphoglycerate, inorganic phosphate is released and produces 3-phosphoglyceric acid, which immediately loses a proton from its carboxylic ion and produces 3-phosphoglycerate. Therefore, again you can say that the product which is produced undergoes converted into other product, and the 3-phosphoglycerate which is produced undergoes resonance stability. So here you can see that inorganic phosphate get resonance stabilized, 2-phosphoglycerate get resonance stability, as well as the immediate product of this 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. So these three factors make this process a very favorable process. The other high energy compound is thioesters. When you are saying thiol, that means we are involving sulfur. So thioester have sulfur atom replacing usual oxygen in the ester bond, and they have very high free energy of hydrolysis. The thioester group containing compounds are acetyl coenzyme. So acyl group in this compound is activated by transacylation, condensation, or oxidation reduction reactions. Thioester undergoes much less resonance stability as compared to other oxygen ester. The difference in the free energy between the reactant and its hydrolyzed product, which are resonance stabilized, is greater for thioester than its comparable oxygen esters. In both the cases, hydrolysis of esters generate a carboxylic acid which can ionize and assume a several resonance form. Together these factors makes this process favorable with a delta G of minus 31 kilojoule per mole. So students, let's now summarize what we have learned from this module. So in the present module, we have discussed the different form of NTPs and their energy contribution. What are the different form of NTPs? We have discussed about ATPs, GTPs, CTPs, TTPs and UTPs. So these all NTPs, if you know that they all have 3-phosphate, and we have discussed when the NTP breaking into its product like NDP and inorganic phosphate, they all have same delta G change. So if they have all same delta G change, then why different enzymes require different NTPs? So we have also discussed, because of the presence of NTP binding pocket in the respective enzymes, they have different requirements. And we have also discussed, if we have an NTP binding pocket here and it has a particular conformation, then it will bind to a specific nucleotide. For example, if your pocket is designed in such a way that it will bind to the ATP, then it will only binds to ATP. Because if ATP will not bind to that NTP binding pocket, then it is not going to adopt the conformation which is required for the catalysis. Similarly, if GTP is required for the binding pocket, then only GTP can bind to the pocket, no other NTP can bind. At the same time, different enzyme require a specific conformation in the active site, which is important for catalysis. We have also discussed high energy compounds such as phosphoester linkages containing compounds, for example phosphoenolpyruvate. So we have discussed how your phosphoenolpyruvate is going to produce energy by having its hydrolysis with delta G negative. We have also discussed some acyl phosphates such as 1,3-bisphosphoglycerate, and we have discussed why this 1,3-bisphosphoglycerate is going to have delta G negative or delta G favorable. We have also discussed what is the significances of thioesters such as acetyl coenzyme A for the ATP production, or we have also discussed how acetyl coenzyme A itself have delta G negative, and that will help for a favorable process, that will help to make metabolic reaction favorable. And we have also discussed their energy production mechanisms. Similarly, we have discussed that hydrolysis reaction of all these high energy compounds are large and negative standard free energy change. We have also discussed, in all the cases, either it is NTPs, either it is high energy compounds, the product are more stable than reactant. Why this is more stable? We have discussed elaborately by taking different examples, but in conclusion you can say the first reason why the product are more stable, the first reason is the bond strain in the reactant due to electrostatic repulsion is relieved by charge separation. This is there in NTPs and any other high energy compounds. We have discussed the second point why they have more negative free energy change is the product stabilization. So product which is formed is stabilized by ionization, as you have seen in ATPs hydrolysis, acyl phosphate hydrolysis and thioester hydrolysis. We have also discussed why sometimes products are stabilized by isomerization or resonance stabilization. In isomerization, you have seen it into the phosphoenolpyruvate. The resonance stabilization you have seen in NTPs. We have also discussed how your product is stabilized by resonance and creating a stable product, which is seen in phosphocreatine, which release creatine and any other molecules. We have also discussed how your acyl phosphate releases carboxylate ion and make the process favorable. We have also discussed about the phosphate release from anhydrides and ester linkages. So, in this way, we have discussed elaborately why NTP is energy currency of the cell, why different enzyme require different NTPs, why high energy compounds produces energy, and what is their biological significance. So, therefore, you can say in this module we have explained different high energy compounds that is found in the body, their role in the metabolism, their role in the functioning of human body. Thank you.